Good day. Welcome back to part 2, talking about genetic engineering again today. I pulled up a picture of these glow-in-the-dark pigs to illustrate again what the possibilities of genetic engineering. I know this is kind of somewhat silly, but there's so many things you can do with it. It's pretty incredible. All right, so last time we talked about kind of getting to the point where we have a target gene and something that we want to edit and put into another organism. Today I want to go through some of the techniques in which scientists can add the gene into an organism. So that's step 3. How are we going to insert this gene? 3 different ways really to look at this. We talked about the heat shock bacteria transformation a couple of weeks ago, where you have your target DNA or plasmid sequence made, and it's in a solution with the bacteria, and you cool it down for a little while, and then you put it into a warm bath and then ice bath, and that'll allow the bacteria to open up its pores and possibly let that target gene into it. And this is another one where they can kind of shock the solution, and that kind of opens up the pores so that the bacteria can uptake that DNA. And I believe they use this to change the bacteria so they can make insulin. That's one of the methods they did that. My primary focus today is going to be on these 2, the viruses, the viral vector, and this new technique, CRISPR-Cas9, which is pretty cool and very new. All right, so the gene therapy is kind of what I was looking at as somewhat of a focus for this. There's so many different aspects of this. And scientists have been looking at gene therapy for a while, which is changing the DNA of a person with a genetic disorder. The goal of that would be to cure genetic disorder, right? You want to go into those cells and change the DNA. And there's a number of problems with this. One, most genetic diseases, they know what gene is affected and why it's defective. But getting into the cells, and there's probably millions of, every cell in your body has that same error in it. So you need to target specific cells and change the DNA. So you can probably make the gene and make a corrective gene, but getting it into and changing it is the issue. And the first thing that they thought of, well, why don't we use viruses? Viruses can get into cells. We know that. We know a lot about viruses. Viruses are very specific to the cell types that they infect. Coronavirus typically infects just kind of the upper respiratory tract, some of those cells. And if you have a specific disease associated with a specific cell, you can maybe design a virus and have it inject modified DNA into that cell and have it get into the nucleus. A lot of assumptions there. The virus you're going to pick is going to be through the lysogenic cycle. Remember, the lysogenic cycle is where the virus gets in and then copies its DNA and genome into the cell's genome. So by doing that, you would have a copy of the good gene potentially in that cell. A couple problems here. Well, it's hard to control. You don't know exactly what cells are going to be infected by the virus and what aren't going to be infected by the virus. You have your immune system kind of to control, to watch out for. It's going to possibly see it as an invader and attack it. And after that, you don't know exactly where in the genome it's going to insert it. So there's a lot of unknowns with this. So even if you get a copy of the gene into the cell, who knows if the cell is going to use it and transcribe it, translate it into the protein you need. So the viruses, the other issue with viruses is finding the right one, and you obviously have to change the virus so that's not going to be causing you harm or going into the lytic cycle and destroying your cells or changing your DNA other than what it's supposed to. And viruses, there's that risk of mutation there as well. And they have used viruses as a viral vector, and they have had some success with gene therapy in the early 2000s. But again, with that immune system, it's hard to predict what will happen. And unfortunately, someone did die from the viral vector due to allergic reaction. So So there is some obvious drawbacks there. The more recent tool is CRISPR-Cas9, which is a gene editing tool, and it allows you to cut and paste DNA. You can cut out the bad DNA and then paste in pretty much the exact sequence you want. It's pretty effective, it's pretty efficient, and it's pretty successful. I'm going to try something different and kind of do a video within a video. It's a short 1-minute video that kind of does a better job explaining it than I could. And there's other resources out there. We'll get to that in the applications here in a second.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
So basically, CRISPR is an enzyme kind of complex with your nucleic acid, and it goes in, scans the DNA, finds the proper sequence that you're looking for that you pre-programmed into the Cas9 enzyme, and then it will cut out the selected DNA sequence, and through some techniques then you can replace it with what you want. It's very effective, very efficient. One of the issues is getting it into the cell. That's one of the problems here. How do you target a specific cell? But they have been able to do a number of different things. What can you use it for? And one of the uses here, you can see a dog. They took, I think these are beagles, and they inserted a gene that doubled or tripled the myosin gene, which is for muscle. And you have the poor old scrawny dog, and you got Mr. Muscles over here. So they did that. They were able to make micro pigs. I know they made the pigs in China. I'm not sure if you can purchase them in the United States. I think they were for pets, micro pigs. The last couple of years, they made pigs that produce less fat than other pigs. One of the earlier uses was to make cattle without horns. So what they would normally do is once they're born and the horns start to grow, because they're kept in close quarters, they would burn the horn, that part of the head where the horns are. They'd have to burn it and scar it so that the horns would grow. And I believe they were able to knock out this gene, and now they have cattle that don't have horns, so they don't have to burn them and hurt them. And then one of the very first one is this. You may look at your yogurt bacteria. Your yogurt, it says it has active cultures in it, and those are live bacteria. And you have these with dairy products, bacteria that can be in there. They use it to grow and to make your food. But there will sometimes be viruses that can infect these particular bacteria cells, and you can decimate entire population of bacteria. And financially, it can hurt companies that are making these particular dairy products. If your whole bacteria population gets wiped out. So they've been able to use Cas9 CRISPR to help, like I saw the term vaccinate, but really give the bacteria tools to kill the virus before it affects it. There are more out there, and it's you've got plants that are being affected by this, all sorts of things, and obviously there's some ethical issues involved, as people are going to start thinking about using this on humans. Like just last month, they had I think they were trying to cure a blind guy or some eye disease. They injected something into some guy's eye with a CRISPR edited solution of some kind in the hopes of changing his DNA. But there's a lot of promise here. With good, there can be bad. So I'm sure there'll be some people doing things they should with this technology. If you're interested more about this, I read this book, Kraken Creation, a number of summers ago. It was a pretty quick read. It goes into more detail. I've got a lot of information there. Kind of goes through some of the ethics involved and the questions being dealt with. And you got time. So this is a good one. And then this video, which I'm not going to play for, it's a little bit longer. A little bit more technical than the last video, but still reachable for those who want more on this. And I'll see if I can't get you the link to this video. But again, the CRISPR is a pretty cool technique for gene editing, and hopefully, making cool things and possibly curing genetic diseases. A lot of novel things you can do with that. All right, enjoy your day.